our first in a new series of GDC webinars we're going to be giving every month. Today's topic is really just an overview of a lot of different features of the Genomic Data Commons. So we have a lot of things to cover. In subsequent webinars, we will dive into all of these different topics in more detail. So yeah, today is really just to serve as an overview introduction. So my name is Michael Fitzsimons. I'm the Director of User Services and Outreach at the Center for Translational Data Science at the University of Chicago for the GDC project. And I'm going to be presenting with my colleague, Colin Reed, who is a Senior Scientific Support Analyst for the GDC. And then just a couple bookkeeping comments. As was already mentioned, we will be recording these. You will be muted by default when you join. If you have questions, please type them in the chat window, and after the initial presentation and demo, we'll be sure to go down and answer any questions that have come in. So with that, today, I'll just first introduce what is the GDC, talk a little bit about the data that is present in the GDC and about the bioinformatics harmonization process. Then I'm going to turn it over to Colin to give an introduction to the different GDC applications, including some demos, talk to you a little bit about resources, and then we'll answer some questions. Okay, so what is the GDC? I like to describe the GDC as a repository of harmonized clinical, biospecimen, and molecular data from cancer genomic studies. Now, really critically, the data is processed using a common set of bioinformatics pipelines and organized using a standard data model. What this allows you to do is to compare data across projects, which, as many of you might know, can be really complicated since data may be processed differently, may use different clinical and biospecimen terminology, and so making those comparisons can be challenging. So that's really one of the core missions, is to have the data in a single place where it looks the same and has been processed using similar pipelines. Another aspect of that is also sort of a feature of the GDC is that we are processing the data before you see it. So you don't have to download all of these raw sequencing files. In many cases, you can download these relatively small VCF or RNA-seq expression files, which also makes analysis simpler. So the GDC also is continuing to add new data. So users can request to submit data to the GDC, which eventually gets released to the community. Also, data can be controlled or open access. Authentication is based on ERA Commons, and authorization is based on dbGaP. So the image on the right so generally I was talking about, with respect to all projects, on the image on the right kind of shows you how the GDC fits into the NCI's genomic characterization pipeline. So for certain NCI projects, the data originates at a tissue source site. The biospecimens are handled at a biospecimen core resource and then sequenced at what's called the genome characterization centers. Those data are then uploaded to the GDC, which are then analyzed by genome data analysis networks and eventually released to the research community. So that's a little bit about what the GDC does and is, but what kind of data do you find in the GDC? So this is DNA, RNA, microRNA sequence data. All the data is aligned to HG38. You're also going to find downstream analysis files, as I already mentioned. And these are the smaller files that are a little bit more digestible. These are variant calls, expression data, copy number variation data, and others. You're also going to find clinical data. So this is information about the diagnosis, the patient demographic, patient treatment, and any follow-up data that they might have been gathered during the course of their treatment or follow-up studies. We're also tracking sample and aliquot information. So this is information about the tissue sample that was gathered and how it was processed in the lab that then led to these data. In many cases, we also have tissue slide images that we can share with the community. So I wanted to give you just the briefest of overviews about the data harmonization pipelines. So the first step when we get raw sequencing data, really regardless of the experimental strategy, is for alignment. So all the alignments are performed against HG38. So all of the alignments in the GDC can be compared to each other. And here is, again, just a couple quick overview of some of the different pipelines. So the first is the DNA-seq variant analysis pipeline. So what this is doing is characterizing somatic mutations by comparing the alignments for the tumor and normal samples. We actually have 5 different algorithms for the variant calling that are used to call these tumor-normal comparisons. We do have a tumor-only variant calling pipeline as well. So for RNA-seq, what this is doing is quantifying protein coding gene expression based on the number of reads aligned to each gene. And microRNA-seq is a similar pipeline in that we're looking at the alignment and then counting the number of reads that align to different microRNA genes in the genome. So that was some of our primary pipelines, but we also have pipelines for copy number variation, for processing methylation arrays. We have ATAC-seq alignments in the portal, and coming soon will be RPPA protein expression data, as well as microsatellite instability data. So those are both coming to the GDC soon. And with that, I'm going to turn this over to my colleague Colin, who's going to introduce many of the different GDC applications.
Hi, everyone. As Michael said, I'm going to talk about the GDC applications and interfaces in a little bit more detail and give a little bit of demos on some of these. Let's see. So we've got a number of different tools for review and exploration of data within the GDC, and you can see some of these here. We've got the GDC data portal, the GDC website and submission portal, and basically some of these tools let you query, analyze, and download the data, as well as give options for submitters to upload and validate the data as well. And we also provide some documentation on our GDC website and documentation site. So I'm going to walk through, firstly, the GDC data portal, which is kind of the main centerpiece of the GDC and the way that people can search and explore the data that we have. So the link is in the bottom right corner. It's the portal.gdc.cancer.gov. And anyone is free to go to this website and look at the data that we've got. And you can browse data by project, files, and cases. And as Michael mentioned, we also visualize a lot of this clinical and biospecimen data, so you don't have to download it. And we provide some exploration tools to visualize the data. And users can search and select cohorts and add the data to download outside for analysis outside of the data portal. And I can show you a slide, but it's probably better to just demonstrate some of this functionality. So I'll do that right now. So I'll close this out and go to the browser. I'll do Google Chrome. So this is what the GDC portal looks like. And it has a number of key features for exploring and visualizing the data of interest. And before I jump into some of these sections on the portal, I want to mention in the top right corner, we have this button here, GDC apps. And these are all the apps that were on that main slide that I introduced with. So if you want to learn more about that, you can go to the GDC portal website and kind of jump right into those different apps. So there's a number of ways to look at the data. We've got projects, exploration, analysis, and repository. I'll step through these very briefly right now. So the first thing you can do is explore kind of what projects we have in the GDC. The projects page lists out all the projects, and you can see what data is available for each of these. And we provide some visualizations at the top for some of the data that we've analyzed. But the primary way to kind of explore the data in the GDC is using the exploration page. And on the left, you see a number of facets that you can search by. So you can choose primary site, program. And what you can do is, if I type in pancreas, this will filter all the cases that are in the GDC by that. And then let's say I'll choose TCGA. And you can see on the right, this all gets updated. And these are the cases within that cancer study. And you can see some more information here. And each of these tabs on the right summarize the different data within that cohort of patients. So if I click on some of these, so genes, you'll be able to see some helpful visualizations like the distribution of the most frequently mutated genes, survival plot, and then what those genes are at the bottom. Same with the mutations. You can see survival and a list of all the mutations within that cohort that we've already analyzed that you don't have to do mutation calling on. So another thing I wanted to point out is we talked about clinical and biospecimen data. So some of these cases here, if I click on one of the case here, you'll be able to see a summary of the case as well as what kind of clinical and biospecimen data that we have. And you can download that from this site here. The other thing you can do is with the clinical and biospecimen data, you can add those as filters on the left. So I'm going to add a case filter to choose tumor stage, which is one of the clinical data elements that we've indexed in the GDC. I'll choose stage 2A, and that again filters and updates the information that you'll see on the right. And what you can do is after you've explored this, we have a couple analysis tools and more coming on the way for reviewing the cohorts that you've created on the exploration page. So I'll just launch a demo here. And what this is doing is you can create a cohort of patients and compare some statistics based on the differences between the two cohorts. You can see survival based on the cohorts and some other clinical information. But we'll talk a little bit more about that in some upcoming webinars. Let's see. So if I go back to the exploration page, what you can do, the last section, if you want to download the raw data for these patients or any of the information that's presented here, what you can do is go to the repository page, view the files, and add some filters based on the type of experimental strategy that you're interested in. So let's say I'm interested in RNA-seq data. I can add that filter here. And then that will, again, filter all the results on the right. Let's say I'm interested in HTC count data, expression data, and again, you can see that all getting updated on the right. And once you've done exploration and you want to download the data, what you can do is add the files to your cart, and then this cart page will summarize all the files you want to download. And then this is actually a good segue into the next tool that I'm going to talk about, which is downloading the data from the GDC. So I'll switch back to the PowerPoint and review one of the slides here. Oops, I started at the beginning. Let's see. Start from current slide. There we go. Nope, that's not it. There we go. All right, so the GDC data transfer tool is the preferred way of downloading data from the GDC. It's a command line tool, and you can use it to download a file or a group of files. And you can also provide it a token file for the controlled access data. Like if you wanted raw sequencing data, you would add that as an option for downloading. And what users can do is you can download a manifest from that page that I showed on the previous section and run a command to start the download. So that's what I'll do in this next demo, is I'll actually just demonstrate using the GDC data transfer tool. So if we go back here, one thing I did want to mention is from this page, you can also download all the clinical and biospecimen data for those files. But for bigger downloads, more than 5 gigabytes, you're not going to want to wait on the data portal for terabytes of data to download. So you would download a manifest file, and what that does is it's basically just a list of all the UUIDs of the files. And then what you can do is open up your command line prompt if you've downloaded the GDC client, and then run a command to start a download using the manifest that you had just downloaded. And this is all really well documented on our documentation site, but I'll just run a command just to to show you briefly how you would do that. So GDC client download, manifest option, GDC, there we go. And then, yeah, that was pretty simple. So you would just download all the files that you're interested in. It will populate all those files there. So I'll jump back to the PowerPoint to keep going here. So I'll hit the right button this time, play from current slide. There we go. So we have a command line tool as well as a user interface tool to make it a little bit more user friendly for people to download data from the GDC. And I just did the demo there. So the next thing I want to talk about here is the GDC API. And the GDC API is a very powerful part of the GDC, as most of the data, a lot of the data is open access. We provide a programmatic interface to query, download, and submit the data. So the API lets users do that, and then it powers all the apps that we've currently got here, the data transfer tool, the data portal, and you can build your own apps using the API, and it uses standard HTTP request methods and JSON as a communication format to pull data from the GDC. And this is what the structure of an API call looks like, just very briefly. It's api.gdc.cancer.gov, and we provide a number of endpoints that allows people to pull data from the GDC. So here's a list of some of those cases, files. We have BAM slicing as an option if you wanted to get only a part of a BAM file. And then we've also got some additional endpoints for some of the more advanced visualization features for genes and survival analysis and things like that. All of the things that you see on the portal already, you can kind of tap into and create your own requests. So here's just some sample API requests if you want specific information about a file, if you want specific information about a case. And we've got more advanced options that you can append to an API request to get, for example, specific fields in our data, or additional filters that you want if you just want to bring back particular data that you want to retrieve. And there's a number of ways to run an API request. The quite simplest is just to copy and paste the URL into a browser, and it'll just bring back results for you. Or you can use some of the third-party HTTP client tools to bring back data, or get more advanced and build your own application using Python or JavaScript or something like that. So the next thing I wanted to talk about is the GDC data submission process. Let's see here. So how do we get data into the GDC and make it available for researchers? We have a number of tools for doing that. And we allow researchers to submit data for harmonization and eventual release to the research community. And the submission of the data adheres to the NIH-NCI genomic data sharing policies. And we require that organizations register their projects in dbGaP. That kind of gets the ball rolling for the submission process. And in general, we accept biospecimen, clinical, and genomic data from human research subjects. And this process is really well documented on our website. We'll definitely do another webinar that talks more in detail about this whole process. But the first thing you can do if you want to get started is to fill out a request form. We've got a website to add some information and begin the process of data submission if you'd like your data to appear in the GDC. So to facilitate the submission process for submitters and people that are wanting to get data in the GDC, we have a submission portal, kind of like the GDC portal, that enables people to submit their data to the GDC. So it has a project dashboard to review all the data that's currently been submitted, submission wizard to help people in uploading and validating the data that they're going to provide to the GDC, and then a browser here to view the data. And again, we've got this all really well documented on our documentation site, and it's a really important piece of the GDC. So the next thing I'm going to cover is the data model, just briefly. And what that does is we have a model for how all the different data fits within the GDC, and this is the diagram for this, and it defines all the data within the GDC, and each of those ovals there kind of refers to the different entities within the GDC, so cases, BAM files, etc. And then the lines between each of those entities defines the relationships between those. So a case can have a sample taken and then the sample can be sequenced, et cetera. And what that helps us do is organize the data, and also to supplement this, we have a data dictionary that defines all the different data elements within the data model, and that can be found here. And the best thing to do for this, actually I'll cover this slide and I'll show you a really brief demo, but as I said, the data dictionary provides a list of all the elements and then the relationships between all those elements. And we use a standard controlled vocabulary for a lot of the properties, CADSR, NCIT codes, ICD. And what I'm going to do is just really briefly show you what the data dictionary looks like. I'll navigate to this website. Let's see. All right, so this is our documentation site, and if it loads properly, great. So this defines all the data that you would see in the data portal or the submission portal if you're a submitter. And I'll just click on one of these. So we've got case, patient, clinical data. So if I click on diagnosis, you'll see a summary of what that node looks like, data from an investigation, analysis, and then this is the most important part, is all the properties that can be submitted are listed below, along with the acceptable values for each of those properties. And then we have links to those standard vocabulary that I had mentioned in the slide before that lets you understand what we mean by a specific term in the data dictionary. Let's see, so I will jump back to the PowerPoint. So yeah, the data dictionary is fundamental to organizing the data and the data model, and then basically defines what you would see in the portal, et cetera. So the last slide that I want to cover here is talking about the GDC community tools. So I showed you just a very brief overview of all the apps that we've developed in-house for the GDC. But because we have an API, because we're open source in a lot of respects, we have a growing list of externally developed tools that have been made for the GDC. And they're built in different languages, R, Bioconductor, and Python. And we have a website where you can go to learn more about some of these community tools. And these are some example tools that have been created, the Genomic Data Commons, Sesame. We have some also GDC tools for downloading RNA-seq data and organizing some of the clinical data that you see in the GDC. So I think that pretty much wraps up an overview of all the tools. We're going to break a lot of that out into subsequent webinars. And then I'm actually going to turn this back to Michael to kind of wrap things up and go from there.
Great. Thanks for the overview, Colin. One thing I did want to mention specifically for the next webinar, this will likely focus on clinical visualization. So looking at the different features that we have for visualizing variants and exploring the data in the GDC. So we'll focus a lot more in the next webinar on the data portal. Today we gave you, an overview of everything, but we can really dive into some of those other features in the data portal exploration page in the next webinar, which we'll be sending out an announcement soon on how to access that. So this slide is really just to round out the presentation and show you some of the resources that are available for you. It includes, all of the different applications that Colin showed, and really critically is that GDC assistance line there near the bottom. So that will send an email to the user services team where somebody will make sure to answer your question, whether it's about submission, download, clinical data, whatever. So that's pretty much it for the presentation today. I think we can answer some questions now. I see one question. So yeah, please, anybody, if you haven't done so already, type any questions that you have into the chat screen. If you want to learn more about any of the topics we covered today, or if you have a question on something totally different. The first question is, about, so the question is, IGV has a web page embeddable interactive component written in JavaScript. It would be very useful if such visualizations could be incorporated for GDC BAM files. Does GDC have plans to provide such visualization? So yes, it can be very helpful to see how the reads align to the genome at times, especially if you're really investigating, some variant and you want to really understand if there's something, weird going on there or how many reads support it. It can be nice to see that. We don't have that in the GDC right now. It has come up in discussion before. It's not in the immediate plans to offer some, any sort of embedded genome browser into the GDC, but it is a good idea, and I'll pass that along to NCI and the development team to see if that can be prioritized. It is a useful tool. I know everybody uses it. I certainly want to visualize reads at times. I think what I would tell somebody if they had that question, I would probably point them to our BAM slicing tool. So, rather than downloading these huge BAM files to visualize, some tiny window, we actually have a tool in the browser and the API that allows you to slice out, just for an arbitrary region or just for a gene, et cetera. And then you could, pretty easily export that to IGV, but of course having it embedded is even better. Don't see any other questions, but one thing it occurred to me to bring up is some new features that might come out in the GDC soon. So Colin gave you an overview of some of the exploration tools. One thing we're really focused on right now for our release in a few months is to release sort of more advanced tools for exploring clinical data. So right now, you can certainly filter by clinical data, but what we want to give people is the ability to bin numeric values, to look at these distributions and really hopefully get a even more powerful way to subset or create, cohorts to then explore the variants or to download files related to those patients. So that's a new thing that's coming fairly soon. I guess another thing that might be coming, in the near term is we're always adding new data sets. One large one that we that is on our plate to release is the Genie data set from AACR. So that will be a big update to the GDC that'll be coming, not too far down the road. So certainly come back in the next couple months for updates to those exploration tools, and keep checking in for new data releases as well.
Hi, Michael. There was a request for you to demo the BAM slicer. Can you go ahead and do that?
Oh, yes. Yes, we can definitely do that. Are you able to?
Yes, I can jump back to the browser. So let's see. I forget where. Here, I'll give this to Michael.
We're sitting in the same room.
Yeah.
So what you would do to slice BAM files is go into the data repository, and probably you've selected, you have some specific BAM files you're interested in, but I'm just going to dump all the BAM files into a window here. So any BAM file, if you go onto this page, you will be able to slice. BAM files in general are controlled access, so I would need to be logged in. Yeah, so maybe that's not a very exciting demo at the moment, but this is a pretty easy way, and there's a real simple kind of help screen that comes up that tells you how to specify it with either chromosome and absolute coordinates, or you can give gene names as well. So it's pretty easy to do. There's also, if you like, there's API instructions on that doc site as well. And while we're talking about that, let me just point out some of these doc site area. So if you have questions about the API, as Colin said, there's pretty extensive examples. There's a walkthrough of the data portal, although that tends to be, certainly we work to design it so it should be very intuitive, but, if you need more information, this would be the place to get it. For data submission, there's all sorts of helpful information there, especially with respect to the data model that Colin showed, and as well for the data transfer tool. We are already looking at the dictionary viewer, and then the data section gives you really in-depth information about our different pipelines and, in some cases, the different file formats that we've created and distributed. Each of these also have accompanied release notes, so when we have a data release, for example, you can always go here to get, real specific information about what has changed. Are there any other questions from the group? Okay, well, I don't see any other questions, so I think we'll adjourn for today. Thanks, everybody, for attending. Please come back or check back for our subsequent webinars. The next one will be on really diving into the data portal and the exploration section. But we're trying to schedule webinars just about every month now. So we'll be diving into these different topics, such as submission, which will cover the data model in more detail, and other features of the GDC. So thanks so much, everybody. Have a great day. Bye-bye.